Hello everyone, I'm Eliana Ruggiero. I'm actually a postdoc in Chiara Bonini's lab in Milan at San Raffaele Scientific Institute. And today I would like to discuss on how can we use CRISPR for adoptive T-cell therapy in AML. So I would like to start by introducing a little bit the concept of adoptive T-cell therapy, because we know that right now that ACT with the engineered T lymphocytes represents actually the front line of cancer immunotherapy. And in particular, it has provided really great results in B-cell tumors. But what about acute myeloid leukemia? We know for AML that the disease is sensitive to immunotherapy, and we know that can be cured by allogeneic hematopoietic stem cell transplantation. And we know that this is due to the beneficial action provided by the T lymphocytes by the graft versus leukemia effect. Still, we need to consider that graft versus leukemia can be counterbalanced by graft versus host disease. Thus, we need to consider and we need to find a new treatment to prevent this. And that's why we decided to focus on CRISPR. So the idea that we had was to really focus on a new technology to generate engineered T lymphocytes that could be used to specifically recognize tumor cells while sparing the host from the graft versus host disease and from the transplant-related toxicities. And what we did in the lab was to exploit the CRISPR technology to completely and permanently disrupt the endogenous TCR repertoire in T lymphocytes and to completely redirect T cell specificity towards the tumor by employing tumor-specific T cell receptors. Actually, in our setting, the complete disruption of the endogenous TCR repertoire was fundamental because was needed to prevent the generation of mixed TCR heterodimers that could be endowed with new specificity, potentially harmful and unpredictable. In our setting, by applying CRISPR-Cas9, we could then generate engineered T lymphocytes that were able to specifically kill primary leukemic blasts, so taken from patients, not only in vitro, but also in vivo. In fact, we could see that we observed a control of leukemia outgrowth also in murine models. And of importance, we need also to note that all this occurred in the absence of the unwanted recognition of healthy tissues, thus indicating and highlighting the safety profile of the approach. But still, I think that in the AML, CRISPR could add additional benefits to the field. Why? Because we also recently showed that actually inhibitory receptors that are molecules that are involved in the induction of exhaustion mechanism in T lymphocytes are highly expressed by T lymphocytes infiltrating the bone marrow of AML relapsing patients. Thus, again, by employing the CRISPR technology, we could think to disrupt simultaneously not only the endogenous TCR repertoire, but also inhibitory receptors or other negative regulators of T cell function. And by applying all these technologies together, we could then generate living drugs that are able to specifically recognize and eliminate cancer cells, but also able to counteract the immunosuppressive tumor microenvironment.